As its name implies, the Devil's Hole pupfish lives in a truly hellish environment. Confined to a single deep limestone cave in Nevada's Mojave Desert, 263 of them live in water that hovers around 93 degrees Fahrenheit year-round, with food resources so scarce that they are always on the edge of starvation, and with oxygen levels so low that most other fish would die immediately. In a paper published this week in the journal Proceedings of the Royal Society B. University of California, Berkeley, biologists report the first complete genome sequences of 8 pupfish species from the American Southwest, 30 individuals in all, including 8 Devil's Hole pupfish. High levels of inbreeding are associated with a higher risk of extinction, and the inbreeding in the Devil's Hole pupfish is equal to or more severe than levels reported so far in other isolated natural populations, such as the Isle Royale wolves in Michigan, mountain gorillas in Africa and Indian tea. Although we were not able to directly measure fitness, the increased inbreeding in these pupfish likely results in a substantial reduction in fitness. Other pupfish species are also inbred, the researchers found, but only between 10% and 30% of their genomes are identical. This tends to burn in or fix, rather than weed out, harmful mutations, potentially dooming a population to extinction by mutational meltdown. The Devil's Hole pupfish species is currently doing well in the wild and in captive or refuge populations, but such low genetic diversity could spell trouble as the climate changes and human impacts become greater. In the face of these potential threats, the new genome sequences will help scientists and conservationists assess the health of native pupfish populations and potentially intervene in refuge populations to increase the genetic diversity of these species, the Devil's Hole pupfish, in particular. With this new genomic data, there's a lot of potential to look not just at genetic diversity and how these species are related to each other phylogenetically, but also look at inbreeding and mutation load to get an idea of what their current status is, how evolutionary history may have influenced T. About 30 species inhabit warm, salty desert springs and streams in California and Nevada. Martin has studied various pupfish populations, including several on San Salvador Island in the Bahamas, to understand the genetics behind their adaptation to extreme conditions and unusual ecological niches. Part of the question about these declines is whether they may be due to the genetic health of the population, Martin said. Maybe the declines are because there are harmful mutations that have become fixed because the population is so small. The small population is partly a result of human incursions into their habitat, Martin noted. Local ranchers and developers pumped groundwater in the region in the 1960s and 70s that drastically reduced the water level in Devil's Hole, leading to a drop in population levels. Nevertheless, a decline in the 1990s led the wild population to its nadir in 2013 to 35 individuals. The wild population has since recovered, while the refuge population has ballooned to about 400, twice the wild population. Humans are not totally to blame for the lack of genetic diversity in the Devil's Hole pupfish, however. The UC Berkeley researchers also sequenced the genome of a pupfish collected in 1980 and held at the University of Michigan. This implies that the pupfish has likely seen population bottlenecks frequently over hundreds, if not thousands, of years. One result of this, Martin and Tien found, is that 15 genes have disappeared entirely from the Devil's Hole pupfish genome. 5 of them seem to be involved in adaptation to living in low oxygen or hypoxic environments. These deletions are a paradox, Because this is a habitat where you're most exposed to hypoxia, Martin said. It could have something to do with the stability of the habitat over time. But it looks to us like the hypoxia pathway is broken. Once you break one gene, it doesn't really matter if you break additional genes in that regulatory pathway. Thanks for watching. Please subscribe my channel for more videos.